So, hi everybody. For those of you that don't know me, my name is Louise Lavictoire, and welcome to the latest instalment of the FBA's seminar series. We hope you've enjoyed the seminar series so far, and if you've missed anybody's talks, you now can catch up with those on the FBA website. So we'll put the link in the chat box so that anybody who's interested in can get straight to that. And also, if you have any questions for Suzanne during the course of her talk, can you please put them in the chat box and we'll address those at the end. If you're enjoying the seminar series and you're not an FBA member, you can join at fba.org.uk and click on Join Us. But without further ado, I'd like to welcome Professor Suzanne McGowan. Suzanne is Professor of Freshwater Sciences at the University of Nottingham and a Research Associate at the British Geological Survey. She's been a member of the FBA since she was a PhD student and was recently appointed as a new FBA Fellow. So yeah, take it away, Suzanne, when you're ready.
Thanks, Louise, and hi everyone. I'm really delighted to be here. It's great to see some familiar faces. And I was asked to kind of present something that encapsulated my career. And really the underlying theme has been pigments. So I've been working on pigments since I was a PhD student. So I thought I'd use the pigments to kind of underpin the talk. For those who weren't at the, we had an inauguration event a week or two ago, and the really embarrassing photos came out there. So if you're after really embarrassing photos, this is not the talk. It's mostly science, but there's one or two photos. So this, yeah, so I'm going to start off with, I suppose, what pigments am I talking about? Lots of pigments in microbes. So the ones that I mainly focus on are chlorophyll type pigments, which are generally green coloured and used for light harvesting in photosynthesis. And obviously that's the colour that often leads to the characteristic colours on ponds for phytoplankton blooms. But there are lots of other types of pigments as well. So there's a large group of different types of carotenoids, which by and large tend to be orangish or yellowish in colour, towards even reddish. And these are, generally speaking, used to protect organisms from light damage. And this is a nice example of a pigment called oconone, which comes from purple sulfur bacteria. This is when a lake in Mexico had turned over, so it had released all of these anoxic bacteria from the depth of the lake. So you can see you get this purplish reddish colour from those photosynthetic bacteria. So essentially, I'm interested in looking at the pigments that come from photosynthetic organisms. And one of the kind of ways that I look at these is if you think about what's going on in the water column, well, a lot of that stuff ends up in the sediments, and the sediments produce an archive. And if you extract, this is on its side here, these are some vials of the mud that comes from lake sediment cores, and you add solvents, you can see you can get some quite striking different colours depending on the types of pigments you've got. So it can range from sort of greenish, yellowish, orangish to this very sort of bright red colour here. And this is the main thing about pigments is they're quite simple to analyse, fairly standard techniques. So you would use chromatography, high performance liquid chromatography. You can identify them using absorbance spectra. And using a combination of kind of retention times and absorbance spectra, you can use that to quantify the pigments down a sediment core. And very usefully, pigments are biomarkers for specific types of phototrophs. So you can use them as biomarkers for things like siliceous algae, so diatoms, chrysophytes, for cyanobacteria, for cryptophytes and chlorophytes. So you can see we can tell quite a lot about kind of coarse taxonomic groupings using pigments. We can't go down to fine taxonomic resolution, but it does tell us something about the sediment record that's hard to access using other proxies. And really, this is the sort of niche that pigments have carved for themselves within the discipline of paleolimnology, I think, which is lots of things like diatoms, for example. We can look at species level information, whereas the pigments will give us information much more broadly about phototrophic communities in lakes. And because they're quite rapid to analyse, you know, they fill a really important role in understanding lake sediment records via paleolimnology techniques. So I'm going to start off by talking about how pigments are really useful for looking at blue-green blooms. So this is around about the time I started working. This was hot news. I think this is a National Rivers Authority, as it then was, leaflet about blue-green algae, and it was in the news then. I think there were some incidents at Rutland Water. So this was sort of really an area of great concern, and I suppose it's unfortunate to say that things really haven't changed much during that time. We're still concerned about blue-green algae. This is a message here from Mallam Tarn from a year or two ago. But so, you know, a lot of the research that I've undertaken has concerned blue-green blooms, and that's partly because we have pigments that are specific to cyanobacteria, and we can tell how they've changed by looking at the sediment records. So this is really early work. I hardly have any photos from my PhD. This is my PhD site, Whitemere in Shropshire. And I was back there a couple of years ago. And interestingly, I stuck an underwater camera under there, and there were still great big flocks of, I think this is, it looked a bit like a microcystis bloom at the time. So yeah, we've got loads of blue-green blooms still going on. This is from my PhD. I think it's one of the only photos in my PhD. This is Colmere over the road from Whitemere, where there's a blue-green algae warning sign. And this lake district was very famous for undergoing blue-green blooms, largely because of this book by Mary Webb called Precious Bane, which was from the late 1800s, I think. And it described this breaking of the meres, which was when the meres bloomed. So we knew that there was a very strong history of blue-green blooms happening in these lakes. And so that formed the basis for my PhD, which was in Liverpool. And so we went and extracted sediment cores from Whitemere, and we got a 6,500 year record. And essentially, the vast majority of the pigments that we were detecting were from blue-green algae. So this was quite a kind of interesting development, because I suppose at that time there was this vilification of blue-green blooms as being this solely an unnatural feature that was caused by eutrophication. Whereas we've got lakes here that clearly have been experiencing blue-green blooms for a number of years, you know, thousands and thousands of years. So that's not to say that there isn't a problem with blue-green blooms, of course. And so, but it's just to kind of illustrate how we can interrogate those histories on a sort of longer time scale. So I was always really inspired actually by the work at Windermere. So there's obviously this long-term monitoring record which has been managed by the FBA and CEH variously, that has provided a continuous monitoring record since 1945. And around about the time of my PhD, this photo was everywhere. This is, I think, in Lowray Bay, showing the blue-green blooms at Windermere. So again, this was sort of feeding into that narrative of, you know, this problem of blue-green blooms. So what I was kind of interested in looking at was, since 1945, what we knew about Windermere was that the South Basin had become more eutrophic than the North Basin, particularly in respect to soluble reactive phosphorus. And we had this so-called hinge point in 1965, where we got this big departure and increase in phosphorus concentrations, and also this increase in nitrates. And then in terms of the sort of algal response, it was really in the 1980s when these cyanobacterial blooms really started to kick in on the lakes, on the lake, particularly in the South Basin. So what I was interested in doing was seeing if we could kind of look at whether pigments worked essentially in the sediment record, whether recording the same thing as what was being shown by the monitoring record. And these are some pigment extracts from Windermere, and we've got the monitored data here in blue. So this is showing the phytoplankton cyanobacteria. And this is the sediment record of a pigment from Cyanobacteria, Myxoxanthophyll. And well, the big surprise was what we found was around about 1945, very conveniently, was when we saw this, preceding that, a big increase in Myxoxanthophyll, suggesting there was an earlier onset or earlier abundance of cyanobacterial blooms. And if we look at the more complete record, this is the sediment core record from Windermere. What we can see is we've got community changes that really happened around about 1960, where we've got this increase in cyanobacteria. Then we've got another big change around about 1945. Now, of course, that's pretty inconvenient given that the monitoring record started in 1945, but it really gives us a sort of window into some of the longer term changes. And one, you know, the obvious question is, well, why are we getting this big change in this earlier period? What was causing this? So that was the impetus for some of the work that I carried out at the Freshwater Biological Association, using some of the archives from Winifred Pennington. So these are the Tooting archives here. And she'd done a little bit of work looking at when sewage treatment plants were installed in various parts of the lake catchment. And so I followed up on that work by sort of supplementing it with some work in the archives in Cumbria. And then this allowed us to link together the archives with the sediment records to look at what might potentially be causing changes in Windermere through that time. And the main sort of headline here is, in the South Basin, it's really hard to distinguish the different categories of change. So we added meteorological records, we had agricultural land use changes, and we had sewage treatment plants. And we got a big kind of blob of variability that was caused by all of these different drivers. So clearly they were explaining a lot of the variance in the algae, but sewage, agriculture and climate were all kind of important. And that's slightly different results for the North Basin for a number of reasons that I won't go into right now. But this essentially led on to the work that Heather Morehouse, who started a PhD with me several years ago now. So she started to look at the lake basins that drain into Windermere, because obviously Windermere is at the lowest point of a much larger catchment where there are lots of lakes all around it that are all subjected to different kinds of stresses. And we thought, you know, this might be important in explaining or disentangling some of the reasons why Windermere was, why the algae in Windermere was changing. So she took cores from all of these lakes, and these are the sort of headlines from that, which is, there's a heat map here. So more red means a bigger increase in total algae, or you've got the cyanobacteria here, the blue-green algal pigments here, and virtually all of these lakes showed a net increase throughout the Windermere catchment, and in both cyanobacteria and total algae, with the exception of some of these upland sites. And there's a reason for that, in that they're kind of much more influenced by atmospheric deposition and climate, essentially. So Heather was able to do some much more sophisticated analysis than I was able to do on the Windermere, the initial Windermere analysis. So she did a regression tree analysis, which allowed us to look at the importance of different drivers in all of these lakes, and to sort of categorize it using this analysis. And what was really coming out as significant was wastewater treatment with a secondary driver of climate. And if you look at just the lowland lakes, which are the ones where, you know, there's more kind of housing and things around them, the big thing that was coming out of this was wastewater treatment. So essentially, this is telling us that, you know, the installation of, or the rise in tourism and the development of sort of infrastructure to support that was probably really important in the degradation in water quality throughout this catchment. And yeah, this just summarises some of that that changes in variability, so that it categorizes it into, you see in the wastewater treatment here, we're going in order of altitude. So we start with the upland lakes, go into the lowland lakes, and we've got the more of an influence of climate on the upland lakes versus wastewater treatment in the lowland lakes, which makes sense because that's where all of the houses are. But it's what we're finding is it's not just the number of houses that make a difference. It's the way that the wastewater is treated as well. So as you sort of change the types of treatment. So this kind of also led on to using a similar approach, looking across a system in the central Yangtze area. And these are our collaborators from Wuhan and the China University of Geosciences. This is Xu Chen, who's been a collaborator for a number of years. And this here is Linkan Zheng, who I think is in the audience now. Hi, Linkan. And he's just finished his PhD and was examined last week. So yeah, he did very well in his viva. And he's been looking at 6 lakes across the central Yangtze. And he very painstakingly went to all the records that exist since the just before the 1950s in China on agricultural outputs, urban related indicators. So there are things like the number of roads, the amount of concrete used, things like that. So we categorized them into these 2 different categories, and then Z transformed them to make the variance similar. And it essentially shows you that since around about 1950, 1960, you've had this slow rise in agriculture associated with kind of agricultural reform policies and the drive to increase agricultural efficiency since that time. Whereas this the opening up transformation in the 1980s is, well, after the 1990s really is when it started to accelerate, is what's driven the kind of urban change in this region. And this is reflected in the nutrient fluxes in the sediments. This is a heat map. These are the 6 lakes across the Yangtze, the mid-Yangtze, and we've got heat that defines how much total nitrogen or total phosphorus there is in the sediments. And you can see the fluxes of those nutrients has increased, but we've got predominantly an increase in nitrogen at the upper end of the catchment and more phosphorus at the lower end, where there's a stronger influence of urbanization. And this is reflected slightly later on in the types of algae. So these are different algal pigments that have increased in the sediments and have led to a big increase more recently. So just to summarize that a bit more and think about the effects that this has had on those ecosystems in the central Yangtze, we've seen a big collapse in several of these ecosystems. So what you see if you plot these trajectories of change in pigments through time, you can see some have changed quite a lot, but some have really kind of collapsed into a different ecosystem state. It's bad news really for the kind of water quality in the Yangtze lakes, but we were interested in trying to figure out why this is. And if you line up these on a time scale, so this is the time scale at the bottom, we can identify periods of significant change, and you can see that some change very significantly around about the 1980s when we get that sort of start of acceleration in urbanization, whereas some change earlier and are potentially much more likely to be associated with those earlier changes in nutrient fluxes. And Linkan used generalized additive models to try and link again the drivers with the algal responses, and the headlines really of this analysis are that the main drivers were either agricultural or urban drivers, and this is just looking at the PCA scores, the whole algal communities, and we've basically got agriculture was important in those lakes further up the river, whereas we get urbanization becoming more important further down the river. And similar kind of pattern for cyanobacterial pigments only, where we've got more urban influence down towards the bottom of the river. So this really gives an idea of where to focus efforts in terms of, do you deal with the urban inputs or the agricultural inputs by sort of looking at these different drivers. So there's been quite a lot of work done now on pigments, certainly in a lot of these areas through the Northern Hemisphere, and we were able to do a meta-analysis using a similar approach to the work of Heather's here, where we're just looking at trends through time. And you can see the pattern over this sort of northern hemisphere region is of a general increase in cyanobacterial pigments. So despite what I started off saying, which is that cyanobacterial blooms have been around for thousands of years, which undoubtedly they have in some systems, they have undoubtedly also increased markedly and significantly in lots of lakes in the northern hemisphere. So I just want to change tack a little bit and talk about another application of pigments. And this is something that was really sort of inspired by, I had a postdoc position in central Canada in Saskatchewan. There's lots of these beautiful saline lakes. You can see here there's a lot of lake level drawdown here. The lake's very saline because it's a very arid region, and there's some lovely pigments that get kind of washed up on the shores of the lake there. This is Peter Levitt, who hosted me for my postdoc, and is an expert in pigments, so it was great to learn from him. And we spent some time on this lake when we were out in Saskatchewan. This is a saline lake, which is also miromictic. So the idea is you've got this dense saline water at the bottom with a freshwater lid on the top, and because of the density difference, it's very difficult for those two to mix. And when you sample pigments and filter them, you get this kind of pattern where you've got loads of oconome pigments in the bottom there, and not very much at the top. And so what this is telling us is basically you've got very clear waters here, but you've got photosynthetic organisms down here. And because the lake doesn't mix, the waters are anoxic. And this provides a nice environment for these anoxic phototrophic bacteria. So there's two things you need here. You need the clear water so they can get the light, but you also need the anoxic conditions in the bottom so that they've got the anoxia. And this is actually something that's really quite useful for looking at paleoclimate. So just going back to before my time in Canada, I conducted some work in West Greenland. And there were surprisingly really similar systems because we were working in a very continental area of West Greenland where there were closed basin saline lakes again. And we knew that there'd been lake level variability through the Holocene period. So what we thought was happening was when you have drawdown of a lake during warmer conditions, you've got saline conditions, and you get certain types of plankton living there. It seemed to be as you get an increase in lake level, you get different types of benthic algae growing there. These are cyanobacteria. But really when you've got a wetter period that follows on from those dry periods, this is when you get this miromixis forming, and it forms this environment where you get a lot of these purple sulfur bacteria. So sorry about this diagram, it's not the best, but this essentially is an example of how we could use this long, this Holocene record from West Greenland, which we're sampling here. You can see the pigments within this. You can see the sort of reddish colour of the oconome in those lovely striped sediments. And you saw an increase in the oconome as the time goes on, and this is essentially telling us that we had a lake that became saline in this period, and then there were periods of freshwater influx that sealed that lid of freshwater and caused that lake to become miromictic. So it's a useful sort of paleoclimate indicator, but it does require you to know something about the kind of limnology and stratification of the lake. So weirdly, this also reminded me of a lake in the Philippines. so the connection here is really that in these tropical lakes, what tends to happen is because you don't have the seasonal fluctuation in temperatures, you can get unmixed states occurring. And so you get anoxia building up in the bottom waters of these lakes. So this is Lake Yambo, which is one of the 7 lakes of San Pablo in the Philippines. It's a very clear water and clean lake. We tested that. This is Charlie Bridden, who's also finished her thesis and was examined, I think, last week or the week before, I'm losing track of time. And this is her work that I'm just going to give you a flavour of now. So one of the things so this is a nice clean lake. There are some really degraded lakes as well among the 7 lakes of San Pablo, and we thought we noticed there was quite a lot of aquaculture going on in these lakes, and thought this might be one of the reasons why we were seeing these blue-green blooms. And so her PhD was sort of centered around looking at that kind of research question. But this is not sediment core work. This is looking at oxygen in the water, and I think this is really useful at helping us to understand what's going on in the sediments. What it's basically saying is we've got really low oxygen at the bottom of these lakes because they don't readily mix because of their sort of tropical nature. But you can see we've got a shallowing of the oxygenated zone as we go along this increasing intensity of aquaculture. So the question is that a linked thing? Because if you've got more productivity, it leads to more deposition of organic matter in the bottom of the lakes, and that can draw the oxygen down. So that's one potential reason why that's happening. The other thing that we see is we can see pigments that spike at different kind of depths in the water column. So these are the 2 that have very low aquaculture, and then we see pigments in the deep waters coming closer and closer to the surface. So the idea here is what we think might be happening is as you get an increase in eutrophication, you get more algal blooms, you shade out this habitat at the bottom of the lakes, and therefore you change the environment for these deep water bloomers. So interpreting the pigment record from the sediment cores in these lakes is not straightforward. It's not a matter of you add more pigments and with eutrophication and that's your answer. You know, you're changing this deep water environment, which changes what happens in your sediment record. So what seems to be happening, this is a summary of all pigments, and it's all put into a PCA, and these are 6 sediment cores from those lakes. These are the ones that haven't got much aquaculture, and these are the ones that have got more aquaculture. And what seems to be happening is we've got movement along this axis, which is really indicating that there's a lot of autotrophic algal production. So that's a classic eutrophication response. But where we've got more movement in this direction, where the oakenone is, this is suggesting there's changes in the deep water phototrophs. So we've got some movement in the diagonal, suggesting that there's a relationship between increases in algal production and changes in deep water production. But in these lakes too, we've got changes in deep water phototrophs, and there's some indication that even in the absence of aquaculture, changes in temperature are causing differences in the sort of mixing stability in these lakes. So potential for sort of climate change impacts in those undisturbed lakes. Right, I'm just checking the time here. So this is what I wanted to finish with was the sort of idea of brownification of lakes, and I'm not too sold on this term particularly, but I think it encapsulates the idea quite well. So this is the idea that lots of lakes may be becoming more or less brown because of the inputs of dissolved organic carbon, which tends to be brown tinged in colour. And this is an area, I think, where pigments have got the real potential to answer a lot of questions. It's partly because, the pigments are so responsive to the underwater light climate. So algae will very much respond to how much light there is. And you can see, if there's this much brown at the edge of a lake, you're not going to have so much getting down to the bottom of the centre of a deeper lake. So I'm going to give 2 examples of brownification type of effects. So there's a couple of things to know, and as well as affecting the water, the light quantity in lakes, it will also affect the light quality when you add DOC to a lake, so brown coloured DOC. And so if we think about the solar spectrum of so we've got the photosynthetically active radiation sort of range, the visible spectrum, and then we've got the UVR. And UVR radiation is potentially damaging to organisms. So the DOM in water that's coloured is very effective at filtering out UVR radiation and therefore providing a more hospitable environment for organisms up to a certain point. So I'll go into that in a second. If you've got no DOM, which often occurs in high mountain environments, you've got no vegetation. The DOM comes from vegetation, so it's from organic matter. And you've got an environment like this, which is a really, really shallow lake. There's nowhere for anything to hide there. So this is where the type of environment where you get adaptations in organisms. So these are benthic algae that are producing a lot of this pigment called Cytonemin. And this is essentially like a sunscreen, and it helps to protect those algae from UV damage. So this is quite useful as it gives us a sort of barometer of what's going on in terms of the underwater light climate, which again can be linked to these inputs of DOM and brownification. So one of the sort of contexts in which we've used this was to address this idea of Arctic greening, and there's also Arctic browning now. But so the idea is the Arctic gets warmer, and potentially more shrubby vegetation can spread across more areas of the Arctic. Now, in some areas, this has actually led to Arctic browning, where there's areas of vegetation that are dying off. But the general point is that it's evident that there's going to be changes in terrestrial vegetation in response to climate change in the Arctic. And this is probably going to have an impact on lakes. So if we think about the sort of impact it will have, if you think about a lake that's very, very clear, what we would expect is you've got sunlight stimulating your photosynthesizers, which use nutrients, and they're taking up algae. So you've essentially got a system that should be, they're taking up CO2. You've got a system that should be a CO2 sink, and then they get deposited in the sediments. So you should have a system there where you've got photosynthesis exceeds respiration, and you've got a net autotrophic system. Whereas if you've got a really brown lake, you've got lots of DOC and DOM going in, you've got a system where the bacteria and the decomposers love that. They break down the DOM and release inorganic nutrients, and in the process release CO2. So you should have a system that is a net CO2 source. And you've also cut out the ability for photosynthesis because you've got a brown lake, so there's not much light energy around. Now, of course, it's not that simple in real life. So this would be a net heterotrophic system, but probably in reality, systems change through time, and so what you probably end up with is something a little bit in between, where you've got the autotrophs and the decomposers, and you've got the decomposers decomposing various sources from the autotrophs, releasing nutrients and then feeding the autotrophic cycle. And the balance between these two is what's important. You've also got the complication that some algae are mixotrophs, so they can use light energy, or they can use organic carbon sources. So we kind of need to know what the balance between these things are, because it's the gas fluxes that are the real sort of headline story in the Arctic. How's this going to change gas fluxes? And the sort of interesting thing is, because we've got remains from these kinds of things, we can have a look in the sediments to see what we think's changed through time. And in this Arctic system, what we were doing was trying to use past analogues of warmer periods and of changes in different types of vegetation types around different lakes, to work out what's going to happen in terms of autotrophy versus heterotrophy in these lakes. So the Holocene thermal maximum is an obvious sort of warmer period that we could refer to see whether that might be an analogue. So we took long cores from 3 areas of the Arctic: Alaska, where we're in the boreal forest, in southwest Greenland at the other end of the gradient in steppe tundra, and then in an intermediate zone of boreal low alpine tree line in Norway. And we had various timings of vegetation transitions here to different types of degree of vegetativeness, from just herb vegetation, so not much in the catchments, to coniferous forest. So what we did was to merge all the data from all of those cores, standardizing it first, and to work out what the overall patterns were through those vegetation transitions. So we've got a time series, we've got changes through time in individual sites, but what I've done here is put all of that data together to look at changes through those vegetation transitions. And as we might predict, if you look at the amount of UV pigment, so this is a Cytonemin sunscreen pigment, versus relative to the amount of all other algal pigments, it's telling us that algae were producing relatively more sunscreen in those sites where you've got not very much vegetation. So in the herb and dwarf shrub tundra type of environments, you've got these sunscreen pigments. As you go into the boreal forest, you've not got much of those pigments around because the water's brown essentially. So you've got this increase in colour. The effect that this had, this also had an effect on different types of algae. So these would be one group of autotrophs. So we're using diatom pigments here. And you can see here we've got a decrease in the herb tundra, an increase in the dwarf shrub tundra, and then a decline afterwards. And the interpretation here is where you've got very little vegetation cover, there's a potential for UV damage, and that can really suppress algal production. Whereas you've got the kind of Goldilocks area here, you've got the sweet spot where you've got some protection, but you've not got brown water. But here you've got light limitation as you go further into that gradient. And there was some sort of support for this. If we look at this is a group of algae that can potentially be mixotrophs, not always, but cryptophytes. We see this increase as you go into boreal forest regions. You've got an increase in cryptophyte algae, and they could potentially use these lovely brown organic matter sources. So what this enabled us to do then was to construct a conceptual model to work out what happens as you go through these different transition zones into different types of vegetation. We could look at the literature and look at what sorts of DOC ranges we would expect to see in those vegetation zones, and work out, suggest what we thought the drivers were of algal production in those different areas where we've got this area of maximal production in the middle. So we've got this unimodal relationship, but we've also got this increase in mixotrophic algae as we go up that gradient, and potentially we'd have an increase in carbon sink on one side of the diagram and an increasing carbon source, the propensity to be a carbon source. And the other sort of side of the coin, which I've not mentioned so much paradoxically since we're looking at sediments, is what happens with carbon burial. I'm not really talking about burial rates here, but we might expect that there would be increased carbon burial as we go towards this side of the diagram, simply because there's less degradation as you get these darker lakes. It lends itself well to preservation of carbon in sediments. So this is the final sort of brownification story I'm going to talk about, and I'm going to actually go back to Round Loch of Glen Head, because I think it's had an airing in nearly all of the seminars in this series so far, so I didn't want to make it feel left out of mine. And this is really stemming from some of the work that Rick was involved with at UCL. And so Round Loch of Glen Head has been worked on lots and lots in terms of acid deposition. This is the Attenborough Ponds, by the way. This is not Round Loch of Glen Head, but it had a nice pollution photo at the end of it there. So just for some context, the idea is that acid deposition really became a thing with the growth of industrial emissions. Really preceding 1800. It really intensified around about 1940 and reached its maximum in 1970, at which point there was legislation to address some of the pollutants associated with those emissions. And then after 1990, a lot of the work has obviously been looking at recovery from acid deposition. So what I'm kind of interested in terms of the brownification story is that it's really well known that when you get a lake that becomes acidified, it's quite well documented that one of the things that happens is that a lot of the DOM changes in its sort of composition, and the water clarity increases really, really markedly. So you get these incredibly clear, gin clear, acidified lakes. And then, of course, what we've been seeing in the last few decades is likely to be a recovery response to acid deposition when we've seen this brownification of lakes as acid pressures reduce. Now, of course, this lends itself really well to pigments because of this change in the colour of the water, which is really nicely detectable. So we've done some work on the sediment pigment records in Ranloch of Glenhead, and I'm just going to sort of run you through those various stages of acidification and what we think is happening. So things to look at are, we've got different algal groups here: silicious algae, cryptophytes, chlorophytes, cyanobacteria, total algae represented by chlorophyll A. And then we've got this water clarity index here, which is the UV pigments relative to the number of other pigments. So what we get early in the record, before acidification, is this fairly stable baseline where we've got lowish aquatic production. When we've got mild acidification, there's a really obvious response here. So we've got an increase in our water clarity as monitored by this UVR index. And that has the effect of stimulating some algal production because it would expand areas for the benthos, particularly the cyanobacteria, which do really well because they've got these sunscreen pigments. And so, you know, they would do well in that kind of an environment. Then we get to the stage of severe acidification, and what seems to happen based on the pigment record is that we've got a slight suppression of some of these algal groups, and this is fairly well known when, you know, if you've got very, very clear water, you can only really the things that can withstand the UV damage are the things that will succeed more, and we see that the cyanobacteria, which can produce these UV pigments, are the things that really succeed at this time. But what's really very interesting as well is now we're into the recovery period, and you can see that's not really what I would term a recovery at all. We've got, it's great to see, you know, we've got a return of some of these groups here that are sort of recovering their predominance in the record. But there appears to be, so a really different community to what was existing before. Now, when we took these cores, which was in, I think it was in 2009, there was a decline around about here. So it would be actually really interesting to see what's happened since that time. Yeah, there's this increase here. You've got to wonder to what extent other kinds of pollutants like nitrates and things like that are having, you know, is there a fertilization effect there? Okay, so I think, all right, that's not bad timing. Sorry, I was galloping through that because I was worried I had too many slides. So, so just to summarize some of the things that I think pigments are great for is, you know, they fill in some of the gaps that other proxies can't, particularly, you know, the cyanobacterial end of things, which is, you know, it's hard to detect them quickly and simply using other proxies. They're very good at detecting deep water blooms, and this can be really useful for thinking about changes in lake stratification related to climate shifts, for example. They're really good at detecting shifts in lake water clarity, and there are lots of kind of applications in that area related to shifts in DOM and all the things that are going on in that area at the moment. Okay, so yeah, I just wanted to finish up by, we summarised some of the work we've been doing in the Pigment Lab since we set it up in 2008 in Nottingham. And this is where we've been working so far. So I know this looks like an evil world map of domination, but it's just quite nice to put a pin in the map and work out, you know, where you've been in the world. And yeah, so this is, you know, there's lots and lots of questions we can answer with pigments. I have to say, Google Maps kind of shifted everything that way, so we're not working in the ocean, but it gives you an idea of the spread of where we've been working. And I just want to finish off by thanking people. I started writing a list of who to thank, and then I decided I was going to offend someone if I even tried to get anywhere near complete. So you all know who you are. Thank you so much for all of your support. And it'd be great if you've got any questions. Really happy to answer questions.
That's great. Thanks, Suzanne. Yeah, if anybody has any questions, please put them in the chat box and I can make our way through them. I have one to start us off. So you mentioned in the 1960s there was this hinge point in Windermere, but the cyanobacteria blooms didn't come in until the 1980s. Do you think that's because concentrations of nutrients kind of reached that tipping point for cyanobacteria then, or was there a reason for that lag?
So it's hard to answer that, because I think a lot of those cyanobacteria are kind of deep water bloomers, aren't they? There's Cytonemin type ones, and I'm trying to just remember back to sort of Colin Reynolds' work at what he kind of showed with that. What our longer term analysis suggested was that it was to do with flushing rates of the lake. So there was a really strong link with spring rainfall, which seemed to modify whether you got cyanobacterial blooms or not. So, you know, I wouldn't want to speculate on you know what's been found out from the monitoring records, but our work showed that there was this link between it was nutrients from various sources coupled with spring rainfall.
Okay, interesting. Thank you. We've got a question here. Has there been much work done on sediment pigments in large rivers?
So there's been work done on pigments in large rivers. We're just working on some stuff in the Red River in Vietnam at the moment. So we're looking at phytoplankton there. The difficulty in large rivers is if you want to do sediment pigments, you need a depositional environment. So you need to be working with kind of, I don't know, the billabongs or floodplain lakes and things like that. So I did some work when I was up in Canada on the Peace Athabasca Delta in the floodplain lakes there. So very, very shallow floodplain lakes. So, and that was, it basically showed that, you know, there was quite a strong response to when the flood frequency declined in those lakes, you got a kind of drawdown and an expansion in the benthos, so it increased overall lake productivity. So I don't know if you'd call that large rivers, because it's the lakes near the large rivers, which, you know, those are the things that have the sediments in. But we've been monitoring the ceston in the Red River and looking at what's there. It's essentially incredibly turbid, and so there's a very low concentration of actual pigments there because of the light limitation, but there's really clear seasonal variability.
Great, thank you. Another question was, what software do you use or recommend for the Hplc pigment analysis?
So we use ChemStation. I think that's just a standard. I'm not sure if I'd recommend it. It's pretty clunky, and you know that's just standard chromatographic software. So what is probably quite frustrating. So you can set it up so it's got libraries to match spectra with your pigments, and you know, so theoretically you can automatically identify all pigments. In practice, we just don't do that because you can't check on all of those things that might have slightly co-eluted or, you know, just come out at a slightly different retention time. So much to probably all of my students probably hate me for this, but yeah, they go through each spectrum individually, sort of truth checking it to see whether it's the right ID. So I think is that what that question was getting at? I'm guessing.
Yeah, I think so. Yeah. It sounds like there's a gap in the market there to try and create something a bit more appropriate.
It's a bit of a problem in sediments. You get things that are slightly degraded, or you know, they can run into each other when you've got a lot of degradation products. So it's not as, you get beautiful peaks when you're working with fresh plankton. So it's much easier with fresh stuff, but yeah, there's always a little bit of an eye has to be cast over the degraded stuff.
And I guess with the degraded stuff, would you also get degraded DNA, so you couldn't even run sort of DNA analysis next to your
so it's not I mean, it's a different extraction technique. So yeah, you can do that together. So we've been working in with some Swiss collaborators looking at how the two gel together.
Oh, okay.
in some of these Swiss lakes, you've got massive increases in cyanobacteria recently, and it marries together really nicely with the DNA. So, but when you've got a 100-fold increase in cyanobacteria, it's
it's a bit easier. Great. Questions are still coming in. So the next one is, with the Arctic brownification story, did you find or expect different responses in shallow versus deep lakes based on differential sensitivity to high light?
Yeah, so you would totally get different responses in those lakes. What we tried to do, because of the, we could only do a certain number of lakes, so we had to choose lakes that were similar in morphometry. We chose deepish lakes, partly because they're better for collecting good sediments, but not too deep. So yeah, they were about between 15 and 20 meters deep. Smallish lakes, so that they'd have a good amount of relationship with the catchment. If you see, the catchment to lake area ratio was good, so we'd get the sort of signal from the catchment vegetation. And also we wanted ones that were in, so we avoided that continental area that I showed you in West Greenland. It was in the inland bit. We avoided that area because I wanted somewhere that was going to be actually washing DOM into there. So we went further towards the coast where there's more rain there. So yeah, we had to, but yeah, it's difficult actually. It's always a thing in paleo. If you do surveys of lakes, the thing that always comes out as being important in determining your sediment characteristics is lake depth, because it's one of the major drivers of sedimentation. So yeah, so we tried to design that out of the study.
Thank you. Another question. In Windermere, you identified wastewater treatment as influential for Windermere. Did that reflect connections of the existing human population to sewers, or predominantly to an increase in human population density, or both?
Yeah, so that's a good question actually. So we had human population density as one of the drivers. It came out as completely irrelevant, whereas it was all to do with when sewage treatment plants were installed, but also we put in a kind of variable that said when they had upgrades. So some of the earlier sewage inputs into Windermere were untreated sewage, and then you got upgrades where it was essentially discharging mineralized nutrients, little nice fresh nutrients for the algae to live off. And it's one of the ironies of we've improved the sewage treatment, but we've made everything much easier for algae to grow. So
okay. So another one about the brownification. If you're using pigments to estimate the phytoplankton community composition, how do you disentangle the increase or decrease in taxon-specific pigments due to the changes in the community versus changes in the cell pigment content? Okay, so
is that asking about is it asking about the UV pigment? So, or
not specifically.
Oh, I see. Yeah. Okay. I see what this is getting at. Yeah, that's absolutely right. So when you've got changes in the light climate, you'll get overproduction of certain types of pigment. Yeah, relative to what you would get, or if you're trying to protect from light. It's one of the big issues with paleo, which is you can't. But I think you just have to, if you. Yeah, it's possible that could be an issue, but if you look at the changes in some of the community assemblages, it's not just one species. The idea that the whole community would be showing the same pigment level response, it seems unlikely to me. So, but yeah, okay. But undoubtedly it's an issue.
Okay, thank you. Another one, this person is interested in the West Midland meres. What do you think is driving historic blooms of algae or cyanobacteria in the early paleo record, if not enrichment?
So that early part of the record is quite interesting. So where we took those cores from in that lake, was about at 7 meters, and there's a pothole in that lake that we missed at the time. So what happens in that record is it runs out at 6,500 years ago, whereas I think there's been longer records taken since then. But what that shows us is that during the Holocene thermal maximum around this area, there was probably quite a significant lake level drawdown because it was drier conditions, warmer conditions. So I think undoubtedly that had an influence on, reduction in lake flushing rates, which, the cyanobacteria really like that. I think there was probably, at that particular coring site that we're at, I think there was an accentuation of what's going on in terms of, I think there was a lot of benthic algae in that area, to be honest, as well. So, there was cyanobacteria, later on when it kind of increased its lake level, but I think benthic algae were contributing to it. So,
okay.
So yeah, lake level drawdown, warmer conditions.
Great, thank you. Another question, have you ever noticed any correlation between algal assemblages and the amount of tannins potentially reflecting surrounding vegetation in lake waters?
So, no, I've never measured tannins. The reason I'm thinking about it is we're doing some work at the moment in a lake from Malaysia where we're working with Harriet Watt is analysing the composition of the organic matter. And so he's able to say much more about whether it's sort of small, molecular, sort of really easy to decompose things versus these recalcitrant large molecules and things. So I'll probably be able to answer that question next year when we've worked through all that
space then. Yeah, absolutely. And then I think the final one, unless anybody has any further questions, is did you have a chance to look at the one has just come in? Did you have a chance to look at the changes of I've lost my space now because that's moved. Sorry, did you have a chance to look at the cryptophyte pigments under changing environmental conditions? I'm not sure in what study. I'm not sure. Actually, the same person has just asked another question about wastewater treatment and what you would recommend for bloom prevention.
Oh, well, that's the $1 million question, isn't it? I'd be a millionaire now if I knew the answer to that. It's impossible. Yeah, I mean, there's changing the water residence time. There's it's nutrient control, isn't it? Which is hard because there's too many nutrients to control. So it's that's incredibly difficult. So the rising temperatures is not helping either with cyanobacteria. So yeah, I don't know. In terms of the cryptophytes, I mean, maybe I don't know if it's thinking about the sort of the idea that they're potentially mixotrophic, and whether they and undoubtedly they will change the way that they are, the way that they use processed carbon, I suppose, depending on the conditions, I'm guessing.
Well, maybe if that person wants to follow up with Suzanne later, I'm sure you wouldn't mind answering the question if a bit more information was provided.
I can't see who's in the chat. Oh, right, okay, yeah. I've just worked out how to find the chat. Right, yeah. So
I think, well, that's 1 o'clock, and that's quite a few questions you've answered. So thank you very much for that. Everybody's saying thank you for a great talk. So yes, absolutely, thank you, and round of applause if anybody can figure out how to use the reactions button. There we go.
Thanks for coming, everyone.
So
that's my
once
first
again,
at-home seminar, so
well, it was great. Thank you.
A little bit stressed about how to build the energy while sitting at home. But,
The robin helped behind you. The robin helped. Thank you for that. Right. So all that's left to say is thank you, Suzanne, for rounding us off for 2020, and you can join us after Christmas for Rachel Stubbington's talk, which is on the 13th of January, on the topic of the aquatic and terrestrial biodiversity of dynamic temporary rivers. So thank you everyone for joining, and have a great day.